Chemical reactions can occur when reactants collide with sufficient energy to react. The amount of energy that is sufficient for that reaction to occur is called the activation energy, or the E of A. Sometimes a chemical reaction must absorb energy for a reaction to be possible. It could be heat or light, but enzymes are biological catalysts, and a catalyst is anything that can be used to reduce the E of A, the energy of activation. Now, if an enzyme is made of proteins, that is what we know traditionally as an enzyme. However, there is another catalytic category called ribozymes that are made of RNA. So please keep in mind that not all catalytic things are made of proteins. Enzymes are involved in many of the chemical reactions necessary for organisms to live, reproduce, and grow. Digestion, respiration, reproduction, movement, cell regulation. But they're involved because they facilitate chemical reactions. They lower the energy of activation. Therefore, the reaction rate goes up because it takes less energy to do it. But if you look at the graph, the energy between the reactant and the product, the delta G, is unchanged. Now, please remember that these are catalysts, and catalysts are not consumed. They are reusable in your body. Enzymes are very specific. Each particular enzyme can catalyze only one chemical reaction by working on one particular reactant, or the substrate. So when an enzyme and a substrate come together, they form the enzyme-substrate complex. I know it's not a very creative name, but once they're bound together, the enzyme is able to increase the rate of reaction in a process called enzyme catalysis. Enzymes are catalysts. Again, not a very creative name. But once the reaction is complete, the enzyme is released to be able to do its job again, and the substrate has been turned into the products. So this is how an enzyme basically functions. Some points are reversible, some are not, but you should understand the general trend. There are two views regarding the mode of action of enzymes. The lock and key model, which is about from the 1890s, basically says that the enzyme is like a lock into which the substrate fits, like a key. Basically, the shape of the active site and the substrate molecules match. They're complementary to each other. So the enzyme molecule holds the substrate close together, forming that enzyme-substrate complex, and creates the product. Now, this model was modified in the 1950s to say, well, that fit might not be perfect, but proteins are flexible. So what we can do is, as that binding occurs, the groups will align and conform to fit better. So the induced fit model is more recent and has a great deal of support because it explains the stability of the enzyme substrate complex. The lock and key model has a few missing pieces there. Is it possible that they're both right in different situations? Yes. So you do need to be aware of both models. There are many factors that affect reaction rate. The first one is enzyme concentration. So as you can see in the graph, as concentration goes up, reaction rate goes up, but it begins to level. It levels because the substrate, you run out of substrate. Now, the reason why reaction rate goes up is because with more enzymes, you have more frequent collisions, therefore you have more frequent changes, because chemical reactions at its most basic level are just collisions. Substrate concentration is a bit different. Again, as concentration goes up, reaction rate goes up because there's more collisions, but with substrate concentration, you reach a maximum rate of reaction. The enzymes, all of their active sites become full, so they're working at maximum capacity, and so it is saturated. Denaturing enzymes means you are changing their three-dimensional shape so they don't function properly. There are three major ways to do this. You can change the temperature. By increasing temperature, you speed up the collisions and you destroy the molecule. And so you're going to have a very optimal temperature that your body is going to function at properly. So humans is about 98.6. You can also change reaction rate by changing the pH, because those hydrogen ions will cause great deal of problems with enzyme structure, but this is going to be bad at too low or too high, too acidic or too basic. But the optimal pH is going to be different for a stomach enzyme than an enzyme in the liver. The third way is by changing salt concentrations or ion concentrations, because protein shapes depend upon ionic bonds. as part of their tertiary structure. So salinity changes will disrupt those bonds. And bodies don't really deal well with a lot of salt. Example, the Dead Sea is dead because of the extremely high levels of salt in the water. Activators are any compounds that help enzymes function. Prosthetic groups are non-amino acid groups bound to enzymes. It could be a functional group. And FYI, there are a lot more functional groups than the basic ones we learned about earlier on. But you won't really see prosthetic groups as a big component in honors. The other two, however, are much more important. Cofactors, which we commonly know as minerals, are inorganic compounds and ions bound in an enzyme. So up above here we have magnesium in the center of this molecule, which is a chlorophyll. And so magnesium is a cofactor there. Now, coenzymes are different. These guys are organic, but they're not proteins. We typically call these vitamins, and ATP is an example we've talked about before. You also have things like vitamin A, vitamin B, coenzyme A, NAD+, FAD+, all things that we'll talk about more later on. So activators help enzymes function. So all three of these will encourage an enzyme to do what it needs to do. Inhibitors regulate enzyme activity. So sometimes you don't need an enzyme to do all that it's doing. You need to slow it down. Or you get bit by a snake and that venom is an inhibitor to your enzyme, which is why it's deadly. So some inhibitors are reversible, some are not, but there are three basic categories. Competitive inhibitors block the active site, so they compete for that space. If you want to beat a competitive inhibitor, the way to do that is to increase your substrate concentration. So basically you have more chance of colliding with the right thing. Non-competitive inhibitors bind to enzymes away from the active site, somewhere else, and that site is called the allosteric site. So you'll often see this called allosteric inhibition. Now, by binding somewhere else, you disrupt the three-dimensional shape, causing a change in the shape of the active site, a conformational change, which is going to prevent the binding of the substrate. So competitive inhibition and non-competitive inhibition can be seen here. Now, the third one is much more common, and it binds only to the enzyme substrate complex. And it's going to make that enzyme substrate complex take a great deal longer to produce the product and to release itself. So inhibitors, not always a bad thing. Sometimes you need it to regulate enzyme activity, but these are the three major types.